Welcome to MOOC course on Introduction to Proteogenomics. In the last lecture, you were introduced to the effect of mutations on gene expression and how they could alter the signaling pathways. You also taught about how these mutations can lead to the modifications of phosphosites and play a role in downstream signaling processes. Today's lecture by Dr. Carsten Krug is a continuation of the last lecture, and he will cover the use of two important tools, ActiveDB and MIMP.
So we are going to go quickly through the different steps here. I mean, here the idea is to get the principle how it works. So if you don't understand like all the details, that's not crucial, for our hands-on session. But I mean, this is again very similar to what we've just looked at. So you have two sets of phosphosite collection. So one is, okay. So this is actually how we built the model here. So one is all positive kinase phosphosites. So these are known substrates, and then to get like a background data set or like a negative distribution, you choose random negative phosphosites. And then you construct your model, and then you use a Bayesian approach to to determine whether your phosphosite that you are interested in comes from a negative distribution or from the positive distribution. So this is Mani was talking about mixture modeling yesterday, and this is kind of a related approach here. So in this case, amino acid frequencies are calculated by what they call a position-weighted matrices. Again, it's very similar to what we've looked at, but so it's the relative frequency of each amino acid at each position. So again, we have the positions on the x-axis and the amino acids on the y-axis, and they add some error term here. So that's basically the only difference. So from that, you can already calculate these sequence windows here. So in order to calculate the score for a specific phosphosite, they came up with this matrix similarity score, which is actually a concept that has been introduced for genomics sequence analysis. So again, like the principle is you want to calculate a score given a phospho sequence, how likely it is or how similar is it to my position weight matrix here. So this you have for each kinase, and you calculate a score for each phosphosite, how likely or how similar is it to that motif. And it's based on information content. Again, so we don't have to go into too much detail here. But what you do, you calculate the phosphosite, the score for your sequence, you subtract the minimum given your matrix, and you scale it by the range. So in order to build up these Bayesian models, what the software does, it calculates exactly this score for all known binding sites. This is one distribution. This is the blue distribution here. And then in order to get a negative distribution, it takes randomly sampled phosphosites from the human proteome. Right? And then it calculates again all of these scores, and then you end up having a negative distribution. And then for a given, this is what I just said, and then for a given peptide, let's say now you're looking at your peptide that you measured, you get a score which is maybe here, and then using Bayesian theory, we can calculate the probability whether this score is more likely to be derived from that distribution or from that distribution. That's the entire concept. So now with this approach, we can actually calculate kinase substrate specificity. Given a phosphopeptide, how likely it is that it has been calculated by, phosphorylated by this particular kinase or maybe by another one. So now, how do we connect that to mutations? So we basically calculate these specificities for both versions. So one is the wild type phosphosite, and then if there's mutation that happened in this sequence window here, we calculate the same specificity, using the mutated sequence, right? And these can be known cancer mutations from TCGA, or you can upload your own mutations. That's both possible. And so then you basically look for differences in these probabilities. So one is your wild type. Was it very likely that kinase Aurora kinase B phosphorylated this wild type sequence? And now after the mutation, how likely is it that still Aurora B can phosphorylate that? So this is how MIMP calculates the effect or predicts the effect of mutations on these kinase binding motifs. So this is the entire concept of MIMP, and I hope that we will make that work during the hands-on. So this is one example output here that you get from MIMP. So basically this is the mutation that I fed in. So it's an arginine at 1555, which has been mutated into a lysine, affecting this particular gene here. And here you actually see the sequence window. So this is the wild type sequence window. It has this arginine at position -3, and after the mutation, this arginine has been replaced by a lysine. So in this case, we see that the predicted effect is actually a loss of phosphorylation. Exactly. So the motif is gone. So I don't see. So the wild type sequence has a probability of 0.97 to be phosphorylated by AKT1, which apparently recognizes the arginine at -3. After mutation, AKT1 is no longer able to phosphorylate it, to phosphorylate this site. So this is how we read these kind of plots here. And then for the same site, you have other kinase motifs. I mean, what I probably forgot to mention is these kinase motifs are very loose and not very specific. So many kinases share the same motif, or at least parts of the motif, right? As we can see here, the CAMK2A also is able to recognize an arginine. Yeah, loss means AKT cannot phosphorylate this phosphosite anymore. So that's just an example, and we will go through more examples during hands-on, I hope. So here you see, so these are the scores that we've calculated a couple of slides back for the wild type sequence. It's 0.6 for the wild type sequence, and this is for the mutated sequence, almost 0, right? And this difference kind of determines the probability. Further questions before I move on to? This is actual data from a patient. So the question was whether this is real data or like some example data, but this is a TCGA sample that I've used here. Can you use the mic, please?
Because the R and P both have the same nature. So it is highly unlikely that this replacement will lead to loss of phosphorylation. Both are positively charged, and this protein needs one positive charge to balance the negative charge on phosphate. So it is highly unlikely that this R protein replacement will lead to loss of phosphorylation.
So what you're saying is it's very unlikely that this is a loss because it's too unspecific?
No, because if R2A, suppose R2E happens, or R2A or P anything, then that may lead to the loss, but it's unlikely. But that's what I'm asking, whether it's a real data or it's just an example.
I mean, the data that goes into this analysis is real data, but I mean, keep in mind these are all predicted events, right? So we don't know. Of course, yeah. So I mean, using this computational framework, which I just tried to explain to you, we calculate, given this data, we calculate the probability, that this might happen, given our statistical framework. So that's not an experimental proof that this phosphorylation site is actually lost, right? And also, you must know that we don't know these kind of sequence motifs for all kinases. So this model has been trained on 120 kinases or so, right? for which we actually have very highly curated, high quality substrate sites. So it might very likely be that this phosphorylation site after it has been mutated can be recognized by any other kinase that we might not know the motif of, right? Again, so this is all computational, it's all predicted, but it's a way to kind of approach these kind of relationships between mutations and signaling.
Dixon, should be some basis validation, should have validation. So if you have some validation on this prediction, that would be more useful.
Sure, but I'm not the one who's going to the wet lab, so that's your part.
You're getting some values, you're getting some values, so.
Sure. So I mean, what goes in here are phosphosites that I've measured, that has been measured, like in the lab, that have been measured in a sample, in a patient sample in this case, and also we have genomics data that comes from the same patient, right? Somatic mutations in a patient. So these are the data that goes into this analysis. All right, as I already mentioned, so you can access MIMP on a web server. So I encountered a couple of difficulties while trying to run analysis on a web server. So that's why actually I decided to use the R package. Okay, let's move on. So the second tool I briefly want to talk about in the next 10 minutes or so is, it's more like a database. It's not really like a tool, but this database also enables you to upload your own mutations and see what could possibly be the effect of these mutations on phospho-signaling events. Again, so these are all predictions, right? So there's no validation whatsoever. So Active Driver DB is a very well developed, very complete database, proteogenomic database that annotates disease mutations, but also population variants and relates them to PTMs. So we have like two major types of omics data which has been integrated here. So one are like post-translation modification sites, like more than 380,000, and also like more than 3.5 or 3.6 million SNPs. And so basically it also predicts network rewiring impact of mutations, and also it uses actually the MIMP software we just talked about. So it basically comes from the same lab. So there are three types of human genome variation data sets that goes in here. So one is TCGA, the other are disease mutations that come from the ClinFar database, and also mutations from the 1000 Genomes Project. So it uses these publicly available data sets, but also enables the user to upload your own mutation files. And again, this is something that we are trying to do during hands-on. So in terms of proteomics and interaction data, it uses data that is available in several different databases like PhosphoSciBLAST, PhosVML, and the Human Proteome Reference Database. And in terms of PTMs, we are not only looking at phosphorylation here, we're also looking at acetylation, ubiquitination, and methylation. And it also pulls in information of TRAC enzyme interactions and also PTM site-specific protein interactions. So these are all, these information are all passed from publicly available databases and now are gathered in one place. And it's a very nice, very intuitive user interface which enables you, everybody who doesn't have the computational skills to do these kinds of analysis to do those. So what you do is very similar to what we've learned in the previous part of the talk. So we combine genome variation with PTM data, and you basically map SNVs and PTMs to your protein sequence and look what are the non-synonymous events in your protein, and you calculate motifs or using the effect of mutations on the motif around that. But again, so here we are not only looking at phosphorylation but also at other modifications. So and it's very interactive, this entire web page. There's many different ways how to view your data. So one is the sequence view, where you can look at your protein sequence along the x-axis, and then you have like all mutation events or PTM events highlighted in this protein sequence. So you can actually look at distribution of PTMs along the sequence. And it also has a way to build up these interactive network views between proteins, kinases and TRACs and PTMs and so on and so forth. So this is something, again, we are going to explore during the hands-on session. So there's a web server available which works pretty well, and also there's a GitHub page where you can access the tool, you can download it, you can fork it, you can modify it and run it locally on your computer. So to sum up, mutations affect signaling networks. I don't think that there's any doubt that this is not going to happen, or that this is not happening. So there's millions of mutations that have been identified that are associated with cancer, but we still don't know the exact molecular mechanism. So what is the association between phenotype and genotype, or genotype and phenotype? So, and I think that the integrative analysis of these mutations in PTM has a great potential to shed light into these kind of relationships. And here I just pointed out two of these tools. There's many out, and there's also many ongoing efforts. So I know that Bing's group is also working on a very nice tool that uses deep neural networks to look into these kind of relationships. So I just picked these two because they're kind of easy to use and very intuitive to use, and the output is very easy to interpret and so on and so forth. This does not mean that these are the best tools, right? And here I just put a lot of references for all of the papers and tools that I've used here in my talk.
Today you were introduced to two tools, namely MIMP and Active Driver DB, which have been used to explore the mutations and their effects on a few cancers. You are also shown how MIMP predicts the gain or loss of a mutation by calculating a probabilistic score at different locations in a given sequence. ActiveDB is another proteogenomic database that annotates disease mutations and population variants as PTMs. It also provides information about various molecular interactions from publicly available databases. There are many more tools freely available, and you are encouraged to try out using them, and then you will explore many features which could really help you for much better and deeper understanding. In the next lecture, we will look at pathway enrichment using a few widely used tools and softwares. Thank you.